Hi. Hi, good morning. Good evening, everyone. Thank you. I would like to thank the organizers for giving me this opportunity to give this talk. So today, I would like to tell you guys a little bit more about the story that I did as a graduate student when I was in Howard Chang's lab in Stanford. So currently, I'm in the Genome Institute of Singapore. I just started my own lab there about a year ago. So I'd like to tell you about landscape and variation of RNA secondary structure across the human transcriptome. As we all know, in the central dogma, RNA is transcribed from DNA, which is then translated into proteins. And for the longest time, the proteins are known as the main cellular machinery in a cell. The ability for proteins to fold into secondary or tertiary structures is critical for its function. However, RNA can also fold into complex secondary and tertiary structures. So an example of how RNA structure can be important for cellular biology is shown in the P27 mRNA, whereby the 3' UTR of P27 actually folds into a secondary structure, whereby during senescence, quiescence conditions, a microRNA binding site is actually occluded, and then hence the cell remains in quiescence condition. And then when the cell is ready to exit out of quiescence into the cell cycle, into proliferative state, the presence of this RNA binding protein called PUM1 binds to the 3' UTR of this P27, resulting in a structural change in the RNA conformation. And then now this microRNA binding site is now available for binding, and hence there is repression of P27 translation, and hence the cell can actually enter proliferative state. So even though RNA structures have been known to be really important for different aspects of cellular biology, very little RNA structure information is actually currently available. Most of the structure information that we have in the protein database belongs to proteins. So there's more than 76,000 protein crystal structures, but only more than 900 RNA crystal structures. And most of these RNA crystal structures belong to a few really well-studied RNAs, such as ribosomal RNAs, SNO RNAs, tRNAs, etc. Structure information for most of the other cellular RNAs is hugely lacking. So to understand how RNAs actually function in the cell, we really need to have a good understanding of what the structures look like. And then so to be able to do that, we developed a method that's known as parallel analysis of RNA structure, or PARS. And basically, what we do in this method is that we couple enzymatic RNA structure probing to high throughput sequencing. So shown here is the schematic of how we actually go about doing it. We isolate total RNA from the cell, enrich for mRNA, and then we fold the RNAs and treat it with a double-strand nuclease known as RNase V1 that cleave at double-stranded regions. And then upon cleaving, this enzyme leaves a 5' phosphate, which enables the cleavage site to be directly captured and then made into a sequencing library. And then upon sequencing from the 5' end of the 5' end of the adapter, we're able to have an idea of where the double-stranded regions are along the transcriptome. And similarly, we can use a single-strand enzyme known as S1 nuclease that cleave at single-stranded regions along an RNA. And then again, upon single-stranded cleavage and adapter ligation, we can make this into a cDNA library that can be sequenced, and such that upon mapping, we can identify where all the single-stranded regions are along the transcriptome. And then so when we actually have double and single-stranded information together, we can calculate a PAS score, which is basically a likelihood of how, basically a probability of how likely it is to be double or single-stranded at a particular base. And so the higher, the more positive the PAS score is, the more likely it is to be double-stranded. And the lower, the more negative the PAS score is, the more likely it is to be single-stranded. And then so we utilize this method to basically look at the human transcriptome. What do the RNA structures in the human transcriptome look like? Are there any global patterns? How do different factors such as single nucleotide variations affect how RNA structures fold in the human transcriptome? So first, I would like to tell you a little bit more about the global map of RNA segment structures in the human transcriptome. We performed RNA structure probing in a family of 3 individuals: the father, the mother, and the child. And out of this, when we first did this structure probing, the first thing that we wanted to check was that our structure probing makes sense, meaning what should be single-stranded does represent single-stranded, and what should be double-stranded does represent double-stranded. So shown here is one of the slow RNAs, and you can see for most part the loops are single-stranded cleaved, and the stems are double-stranded. And genome-wide RNA structure probing in human transcriptome identify more than about 20,000 transcripts, actually, tens of thousands of mRNAs and a few thousand long non-coding RNAs. And when we average the structure signal across all the transcripts by the translation start or stop codons, we can actually see that in the coding region, we actually have slightly less RNA structure in the coding region than in the 5' UTR or 3' UTRs. And again, we see an increase in accessibility near the translation start codon, and that is correlated with translation efficiency. And then we also see this 3 nucleotide periodicity in the coding region that we have also seen before in yeast that also seems to be correlated with translation efficiency. Interestingly, now we can actually be able to look into specific co-transcriptional or post-transcriptional processes to see if there are any RNA structures that are associated with some of these processes. So shown here is we aligned all the transcripts across the exon-exon junctions to see if there is any structural profile that's associated with splicing. And as shown here, we see an increase in accessibility towards the end of exon 1, immediately before the exon 2. And however, currently we do not really know what is the function of this increase in accessibility. And then similarly now, we can actually begin to go in to look at microRNA binding sites and see if there are any structural regions, structural features that represent real microRNA binding sites, as opposed to falsely predicted microRNA sites. And so to do this, we actually utilized AGO pocket data, which is basically a way to look at RNA regions that are pulled down by the AGO protein, by the AGO enzyme. And what we noticed is that the true AGO-bound microRNA sites are actually more accessible than the non-AGO-bound predicted microRNA sites. And you can see in red are the true AGO-bound, and they're a lot more accessible immediately upstream of the actual microRNA binding site. And we can actually go ahead and sort, put into this microRNA, sort the accessibility of this region into put them into 5 different bins. And we can actually see that the more structured regions are actually less likely to be AGO-bound, and the less structured regions are more likely to be AGO-bound. And actually, when we look at gene expression data, so when we overexpress a microRNA and look at what other targets are actually repressed by this microRNA, we actually see that the sites that are more accessible are actually more likely to be repressed to a greater extent than sites that are less accessible, indicating that accessibility is actually important for microRNA function in vivo. So now I want to move on a little bit into actually how variation or single nucleotide mutations in the genome affect RNA structure. So people have looked at how single nucleotide variations can impact RNA structure computationally. So shown here is one example of the CFTR receptor, which is really important, which is mutated in cystic fibrosis. So shown here is actually a tree nucleotides that deleted in this important protein during cystic fibrosis. And for the longest time, people thought that it was only it was because of this deletion that caused protein misfolding and hence resulting in the severe phenotype of this disease. However, recently it was also discovered that this deletion resulted in a base mutation in the sequence, and then hence when you actually revert this base back to its original sequence, you can restore the secondary structure, resulting in the restoration of the efficiency of translation across this region, and hence a less severe phenotype. So computationally, people have noticed that single nucleotide variation can cause structural changes. Shown here again is a single wild type, how RNA structure in a wild type transcript takes on predominantly one RNA structure, and when you actually undergo mutations, they take on different RNA structures, they shift to different ensembles. However, experimentally, people have not been able to do that very well in a global manner. So shown here are two examples of how the structure profile of father and mother, and you can see that there are 3 different mutations along this region. 2 of the mutations in blue do not cause any structural change, and 1 of the mutation in red results in a large structural change. So these are the structural changing mutations that we're actually looking for, and we can use PARS to identify the structural changing mutations by comparing the structural profiles across the individuals. So shown here is one of the examples whereby we can see that the father undergoes a single base mutation from A to C in the mother, in the 5' UTR of this gene MRPS21. And by sequencing, S1 sequencing, we can see a huge change in the structure profile. That is also we can reproduce by manual footprinting. And you can see here on the right, which is the footprinting gel, that we can see clear structural changes when there is a base single nucleotide change. And then we can also use other ways, other different types of structure probing methods, such as shape or DMS, to validate the same structural change. And you can see that when we use this structure prediction program known as SeqFold, this single base mutation actually causes a dramatic change in RNA structure folding. And when we look across entire transcriptome, we can identify about 1,900 single nucleotide variants that cause structural change. And this is about 15% of all the single nucleotide variants that we identify are expressed between the 3 individuals, the father, mother, and the child, which is a much greater number than previously appreciated. And we can actually validate some of these structural changes by doing allele-specific mapping. So, for example, here, if this father is AA and the mother is CC and the child is AC, then when we map uniquely across either the A allele or the C allele, we should be able to see reads that map only to the A allele or only to the C allele. And you can see when it is actually A allele shown here in the child, when it is mapped across the A allele profile resembles that of the father's profile, and the C allele profile resembles that of the mother's profile, indicating that the structure changes, whatever structure that's present in the father is inherited by the child, and whatever structure changes in the mother is also inherited by the child. And then, so now, because now we have thousands of single nucleotide variants that change structure, now we can actually begin to look into some of the properties and features of these structure changing variants. And what we notice is that when these structure changing variants are found in double-stranded regions, they tend to become single-stranded regions, more single-stranded, and vice versa, as opposed to it becoming more and more double-stranded or more and more single-stranded. And interestingly, the nucleotide content actually influences how the direction of the structural change and how much the structural change occurs. So when the base changes from GC to AT, you can see that actually it tends to be more single-stranded. And when the base changes from AT to GC, it tends to gain double-strandedness. And when it is a homozygous change, the magnitude of the structural change is much, much greater than a heterozygous change. And so now that we have all these structure changing mutations, we have this question: where are they located in the transcriptome? Are they conserved? Do they have any function? So one of the ideas is that if the structure changes occurs in a region that's really important to the organism, then it is not likely to be conserved. It is not likely to be preserved because the cell needs to survive. However, if the structural change occurs if the base change occurs and there is no structural change, if there is no structural change, then it's likely to be tolerated. However, if there is a base change and there's a structural change, and the structural change occurs in a functionally important region, then it's likely to be depleted because the cell really wants to get rid of these structural changing mutants. And what we notice is that these RiboSNitches are actually depleted from functional regions in the transcriptome, meaning that when they are actually in microRNA binding sites or in 3' UTRs, these structural changing variants are actually depleted. So the cells actually do not like to have them in these functional regions because presumably the maintenance of RNA structure in these functional regions are important. And when they are disrupted, it affects the cell's ability to survive. And then, so in summary, I want to tell you that we utilize PARS to identify structures for thousands of human genes, and that we identify certain structural features that are specific to co- or post-transcriptional processes such as splicing and microRNA gene regulation. And we are actually able to see that there is an increase in accessibility that is really, there's a feature of true microRNA binding sites. And this accessibility is important for microRNAs to be able to repress its gene targets. And when we look into different factors that affect RNA structures, one of which is single nucleotide variations, we identified about 15% of these single nucleotide variations can actually cause RNA structural changes. And this is really exciting to us because people have been looking for these single nucleotide variations in all kinds of diseases and different human populations. And although they can identify them, how they function is still predominantly not known. And then so now we can potentially, one potential mechanism through which it can exert its function is actually through changing RNA structure. And then what we notice is that many of these structural changing mutations are actually depleted in functional regions, and indicating that RNA structures are actually important in many of these functional regions, such that when RNA structures are disrupted by these mutations, the cells are less likely to survive, and hence these mutations are likely to be depleted. So for acknowledgements, I would like to thank Howard Chang's lab in Stanford, where a lot of this work is done. And also, currently, I've now moved to the Genome Institute of Singapore, and I've set up my own lab there. And we are actually recruiting. We're looking for postdocs. So interested candidates, please feel free to email me at my email number. So thank you. So I would like to take questions. So one of the questions is there enough past data now that we can take a list of RNA mutations and rank them in terms of their likelihood of being RiboSNitches? Well, yes. So for the 3 individuals that we have performed, we do have enough past data whereby we have a list of how likely it is to change structure. How much for every single mutation, we can calculate the magnitude of structural change that is caused by this mutation. And then we can actually rank them and then hence take the top ranking ones and say that they're more likely to be RiboSNitches. So So that is what we can do. However, because currently we only have 3 individuals, so we can only look at the SNPs or single nucleotide variants that actually express in these 3 individuals. 3 individuals, and then so we need more more sequencing data so that we can actually look at more. We can have a bigger list of structure changing mutations, and then so now that you have let's say you find a mutation that's really interesting to you, you can actually now go in and look into the list and say, is it likely to change structure? Could it be one mechanism that affects gene function? Okay, so another question is, how do you plan on validating the impact of these mutations? So yes, so this is the biggest question, because some of these structure-changing mutations, we really want to know what they are doing. And then so one way that we can begin to look at it is actually to intersect with, for example, like one way is to actually to intersect with RNA sequencing data to see if any of these mutations, structure-changing mutations, are likely to result in gene expression changes. So similar to the concept of eQTLs, but now we're actually able to look at how within this gene, whether any of these, whether there's an association with the mutation with gene expression. Similarly, we can look into whether there's an association of these mutations with ribosome profiling, protein translation, or microRNA regulation, etc. And we can go ahead and mutate these mutations to disrupt, to like revert back these mutations, disrupt the structures to see whether it actually affects gene regulation. And another question is, how do you see different structures of the same RNA in different human cell lines or tissues? So as long as as long as the RNAs are made independently, as long as the libraries are generated independently, we are able to look at different structures of the same RNA. The difficulty lies in if the same RNA take on 2 different conformations within the same cell line, we will not be able to tease apart the different conformations unless there are mutations in between the 2, unless there are mutations in the RNA that allows us to map uniquely across mutations. If it's the same RNA with the same sequence in the same human cell line, and it takes on 2 different conformations, we will not be able to tell. But if it is in different cell lines and we can make different libraries out of it, then we will be able to map back into the RNA and tell you that they have different conformations. So another question is, can the algorithm calculate path score be extended beyond single nucleotide variation? Yes. So we can actually use it to also look at the impact of deletions, for example, or insertions, or bigger mutations, how they impact structure. Have you used a method okay, so another question is, have you used your method to determine how good in silico methods for predicting RNA structure folding are? And perhaps validate which is the best performing? Yeah, so this is actually a pretty good idea. We haven't really gone ahead to do that, but I think it would be really cool to try that. Okay, and then what is another one? Are there any agents available to change RNA structures? So there are multiple agents available to change RNA structures. So I'm not exactly sure what you mean. So one is, of course, like heat. So when you heat it up, it will melt. Another one are cheotrophic agents such as urea. When you add it in, it tends to be more single-stranded. So there are various agents that are available to change RNA structures. Or for example, mutations or okay. So another one, if I understand right, there are some mutations to regulate some mechanisms to regulate mutations. Is it possible in all cells or the regulative mechanisms are present only in some cells to escape the mutations? So yes, so I can imagine that under certain selection pressures, under certain selection pressures, that you can have compensatory mutations, that you can escape the escape the mutation so that you can restore the RNA structure. And this is actually well utilized in so one of the ways to predict RNA structure is actually to use phylogenetic information. And the way that they use it is actually to use covariation changes. So that is across conservation across multiple different species, across over a long time, whereby certain structures are maintained because there are covariation. But within the human population, I think it's a little bit too short to actually be able to see this kind of covariation. We did notice that because originally we were trying to see if single nucleotide variations, if these structural changing mutations tend to be clustered together, or whether these structural changing mutations tend to exist on its own. Because we were wondering whether if you have many mutations, maybe it will exaggerate the phenotype, right? It will make the phenotype even stronger, as opposed to if you only have one mutation. But surprisingly, what we noticed is that these single nucleotide, these mutations that change structure tend to be on its own, whereas mutations that do not change structure tend to be more clustered together. And one of the possibilities is that is because there is covariation. So actually maybe one mutation causes a structural change, which then the second mutation reverses the structural change. And then so we realize that non-structural changing mutations tend to be clustered together. So another question is, have you seen examples of RNA secondary structure mutations that lead to escape by microRNA regulation? So we have not. So there are known examples in the literature whereby, for example, the P27 3' UTR, when it undergoes a structural change, it results in a microRNA site being available or not available. And then so when the microRNA site is actually available, then you can actually, when the microRNA site is actually available, then it is actually, it can actually undergo microRNA regulation, whereas when it is bound, when the microRNA site is actually bound in the double-strand loop, it is not available for microRNA regulation. However, in our own data, we have not been able to show that yet. Excuse me. Okay. Do you think the impact of RiboSNitches could be significant enough that they might be cancer-driving mutations? Do you know of any examples of this? So yes, so we do really think that this could be one way that we can actually, they might actually be driving cancer. I mean, we don't really know. We don't have actual example of this. But for example, some of these structure changing mutations fall within the GWAS loci, loci that not cancer, but other diseases such as Parkinson's disease, for example, or multiple sclerosis. And we don't really know what these mutations are doing. And one of the possibilities is that these mutations are actually causing these diseases by affecting RNA structure. And one way that we can actually go about doing it is actually to do now very systematic studies to look at how these mutations actually change cancer phenotypes. So if we actually do compensatory mutations that change the structures, do you still see the same kind of phenotype in these diseases? So that's one way. And then so one possibility is actually to come up with a huge database, right? Like if we do it now beyond 3 individuals, if we can do it in, 100 individuals across different continents, and then we can actually curate all these mutations that cause structural changes. So now we can actually have this master list whereby any disease, when you have a mutation change from base A to base B, we can now go back to this list and say, oh, actually, this mutation causes a structural change. Maybe we can now look into whether the structural change does anything. So, yeah. So I think that's all the questions I have for now. So unless there are more questions. Okay, so there is one more. So when transcripts are produced, they are exposed step by step and exit from the polymerase. The in vitro folding system of RNA is where full-length RNA exposed. Hence, I'm wondering how valid the in vitro folding is. Okay, this is a long-standing question. People have always been asking to what extent does in vitro folding actually recapitulate what we see in vivo, and we don't actually know. To some extent, it does, because to a large extent, sequence do drive how an RNA is folded, but also a lot of other factors such as in vivo RNA binding proteins affect RNA structure. So now there's other ways in vivo genome-wide RNA structure probing, so people can now systematically go in and see how similar it is, or how different. So I think my time is up. So thank you very much for listening to my talk, and feel free to email me more questions if you have any, and I'll be very happy to answer them. So thank you very much, and bye.